friends, hello students. Once again in the video of the human histology, myself Professor Dr. Raksh Dubey, Professor Department of Human Anatomy, Himalayan Institute of Medical Sciences, Swami Rama Himalayan University, Dehradun. In this continuous lecture series of the human histology understanding, this is the last video of making of the spatial senses. I have chosen the spatial senses for the some voluntary purposes so that give you an idea how a spatial organ could be histologically analyzed, and that give you a more detailed idea of that. In that regard, I have chosen the retina here. I hope that you have understood all these lectures earlier as well to understand it more understand. Well, the retina slide is one of those slides which are very difficult to form under the histology because retina usually get broken out and it required very special technique. It's one of the most finest state of the art we need to make these slides to the retina. And understanding the retina with the HND staining is a little bit injustice with the student because retina cannot be visualized completely under this HND staining or giving its complete histological analysis. In HND staining, though it has a very peculiar pattern of the nucleuses, double rowed nucleuses, multiple double row of the nucleuses, that give you very clear idea that this is retina. Once you see it, you never forget it that this is the retina slide, but if you go into the depth, it's a bit challenging. Let's see this. So I have chosen this slide, and on the right side of this slide, I have given you just a schematic diagram what we are visualizing on another side. What it could be: squamous epithelium, thin epithelium with a big lumen, that is nothing but a capillary. Here we see it, and then there the various connective tissue are present over here, right? And this nucleus of big structured nucleus is basically ganglionic cell, and these are the dendrites of this ganglionic cell. So this is the ganglionic cell layer. Connective tissue is there, then emerged from that connective tissue submerged is the ganglionic cell nuclear layer, and these are the various dendrites which are coming out of this ganglionic cell. From the another side on this area, the axons are coming off this ganglionic structure, and from here the optic nerve is formed from these axons, right? So this is the ganglionic cell layer. Here we see plexiform layer. Since it is on inner plexiform, why it is named as inner plexiform? Because it's inside, not outside, and plexi means network. So here we see the neurons of that ganglionic and the downward bipolar cells are present over here. The mixing is there. That's why this relatively clear zone is named as inner plexiform layer, though it's not at all, nothing is present here. It's a filled up with so many lot of wiring over here and with intermingling wiring. That's why it's named as plexiform layer. Then we have the nuclear layer. It is named the inner nuclear layer. Why it is named inner nuclear? Because present inside, and it is the basically nucleus of the bipolar cell. Here you see that this is cell on the bipolar cell. Another side, one dendroid axons are present. So it one side it meets with the ganglionic cell, and then another side with the photoreceptor cells are present. So after this inner nuclear layer, we see again a gap is present, the clear layer. But this is not clear again. These are the various fiber which are intermingling with this each other at this place, and this is named as outer plexiform layer. So in histology of the retina, you understand the plexiform is the area where if you make it specially stained, if you visualize it again, you see it's a mesh work of or so many wires are present over here and intermingling with each other, while the nucleuses are the actual cell nucleuses which are bigger in comparison to the other ones. Now, inner nuclear layer is photoreceptor cells nucleus, the big nucleuses, more in number, they are adjusting one another. So they are not present in one plane, slightly above one is there and slightly below is the next one is there. So you see the zigzag layer of these nucleuses, and then we have the receptor layer is present, and then outside is the pigmented layer. We already know that retina pigment is present over there, and whenever the light passes through that, all this structure, it is absorbed or reflected from this layer. Now you see this, the light does not come directly as you see in the camera. The light comes into that area and then the signals are formed, but in the retina, light first passes through all these structures, then reaches into the back, and then the signal is generated. This is the difference between the camera where the light first strike on the receptor, but in the retina it is not the truth. It passes through all the layers, and into the deepest layer is the retinal receptor layer where the light is absorbed, and then the signal are generated into the reverse order. So here, and after side, we know that the choroidal layer, of course, and the scleral layer is visible, which are the layers of the eyeball is there, and here we see the retina is present over here. So in the retinal layer, blood vessels, ganglionic cell layer, then the plexiform layer, inside that is named as inner plexiform, then inner nuclear layer, outer plexiform, outer nuclear layer, and the receptor pigment layer and the pigmented layer. These all are the layers are present. Histologically, retina containing 10 layers in itself, and this way you can analyze them. Remember, pink and the blue is the key, especially the blue. If you see a blue dot anywhere in the histology, that means chances are somewhere nucleus is present, and around them you can assume at certain places cytoplasm or the other structures, how they are scattered around this blue dot structure, and this give you an ideation how actually the tissue may look like or what organ actually it is. So this is the way of analyzing the retina. Special cells have some unique anatomy, especially in the histomicroscopic anatomy, so that in first go it's very difficult to analyze them. I am telling you this analyzing technique so that if you meet certain pathology as well in retina, so that you can depict what is the deformity and what sort of cells are present over there, and with on that ground, with that knowledge, you can go and can judge this what could be the disease. So this basic understanding of histology is always required to understand the pathological histology as well in the future. I hope in all these videos you have understood a basic idea of the histological techniques, various tissues, and various component basically of histology, and then in the last slides I have given you how to compile all these basics and make the complex organs. Retina and spatial senses are most complex structures which we see, and if you understand how to analyze them, you can be very successful in the histology, and it's not going to be a nightmare for the students. I thank you all for your patience listening on this series. Maybe see you next time with some another interesting topic. Till then, thank you and goodbye.